This is episode 88 of the Immunology Podcast, Cancer and Inflammatory Diseases with Dr. Roslyn Kemp. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Raud. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Roslyn Kemp from the University of Otago on the podcast to talk about her research on the molecular pathways that control T cells. We also got our usual roundup of recent highlights in the immunology news coming up. But first,
Immunology 2025 is the world's leading annual all-immunology meeting. And in 2025, indeed, it's going to be in Honolulu, Hawaii. Join us from May 3rd to 7th, 2025 to connect, collaborate, advance your career, and access the cutting-edge immunology research. Abstracts open for submission on October 1st, 2024. Learn more at immunology2025.aai.org.
all right.
What up, Jason?
Oh,
Ready for a holiday?
Yeah, I need one. I living the dream of mergers and acquisitions.
Fun. That's all the med school years brought you to this moment, right?
But somehow this is where I ended up. But that's okay.
You should take a sabbatical. You should go somewhere.
They don't, sometimes they have those in industry.
They do?
But yeah, they're pretty uncommon, but they exist.
Well, you could go to Switzerland, to go see the big HQs of your new company.
I could. It'd be exciting to do that.
And I know you're supposed to learn something, like go and learn new things in your sabbatical. So you could learn how to yodel.
I mean, I would do that.
Right?
That sounds awesome.
I'm just going to put it out there.
I mean, yodeling plus dad jokes seems like an awesome combination.
That's going to make you so popular, Jason. You would be the heart of the party. Just do it. Believe me. Just do it. Go to Switzerland.
I'll be the life of the party, then put on the roof.
All right. So, Jason, we're not on sabbatical yet. So I would say, tell me, what's the science you brought in?
All right. I'm going to go with the cool paper first.
That's so mean on the authors of the
It's a great paper, but it's not as immunology as I wanted it to be. I thought it was going to have more immune cells than it did.
Okay.
But it's still cool. It's just, it's more structural. But first up, in the land of very immunology here, from Nature Immunology, published the 12th of September, dual blockade of IL-10 and PD-1 leads to control of SIV viral rebound following analytical treatment interruption. First author, Susan Piero Ribeiro, and last author is Rafik P. Sikali. All right, so SIV, right? Simian HIV. Great models in rhesus monkeys. That's how we study a lot of HIV work. So what they did is they put monkeys on antiretroviral treatment, normal. When you come off of that, your virus levels pop back. But then they had one group that did that and pulled them off. Then they had another group they gave IL-10 blockade to. And this is because that during antiretroviral therapy, there's high levels in plasma of IL-10 and PD-1 in HIV patients. And then the third group, they gave PD-1 blockade and IL-10 blockade. And then at the end, they pulled it all off. And I'll talk a little bit more about all the other little sub-conclusions, but there's one really big conclusion, and I could just be done presenting this and tell you to go read it, is that if in the group that got both blockades, viral levels remain suppressed after they pulled everything off for more than 24 weeks. After you're off antivirals and this anti-IL-10 and PD-1 blockade. So it's creating a durable response and resistance to HIV post-treatment. So that's the top line. Boom. Cool. All right. How? Right. All right. So other things. So what they're seeing is that this blockade strategy leads to induction of inflammatory cytokines, proliferation of effector T cells in the lymph nodes, and notably they hone in on reduced expression of BCL2, which will then lead to proper cell death versus cells persisting and spamming out, CD4 cells spamming out virus. And they're all predictive of this. And there is lower viral load was associated with higher frequency of memory T cells expressing TCF1 and SIV specific CD4 and CD8 cells, unsurprisingly. So what this is doing is mapping out a path and way to treat potential HIV after discontinuation of antiretroviral therapy. But you don't want to, we keep people on it their whole life, that it works, right? It's a miracle of science, one of the ones we have. But what could you do not to have to just keep on everyone all the time? Or how could you make it better, right? Because maybe you need a holiday from the drugs because of the toxicity at some point. And so this is a way to do it. It conditions the cells to A, die if they have the virus, and B, be non-suppressed and kill the virus persistently post-infection or post-removal of the drugs against the HIV infection. So they have single-cell RNA sequencing, they have cytokine mapping, they look at all these subpopulation answers, but in the end, I just gave you the bottom line. It's super cool. I get the therapeutic tingles reading it. It's awesome.
Really nice. So in this model, where does the IL-10 come from? Do they know?
Where it comes from normally, why it's high.
Yeah.
I don't think they get into it in this. It's a known finding in humans that IL-10 is high. It's just high plasma levels exist is what they're talking about. And they talk about how PD-1 expression in myeloid cells drives high IL-10 overall. But they don't know. I don't think they talk in this paper about the where the IL-10 is coming from in HIV model. It's just a known phenomenon.
I mean, this is super interesting. I remember we covered also some months ago a paper in which they were doing, was it an antibody therapy? I forgot what it was. Also, it could give like extended protection against viral reactivation. And it's really good because I know there are certain toxicities related to the regular antiretroviral therapy, right? Well, do you know what is like the most problematic?
There's like GI symptoms and issues with your skin, and there's all types of stuff. And there's a bunch of drugs that affects your ability to go on for other things because of their metabolism.
Yeah.
The earlier ones, I think, cause some kidney disease, but those aren't used anymore.
Yeah, so those are the risks, right? But this is so far only in this macaque model, right? About human translation? What do they think about that?
I mean, the macaque model is the human model in monkeys. It's as close to it's one of the best models we have that matches human to primate because the virus evolved from SIV to HIV.
True.
So, I mean, next step would be just throw this in humans, basically.
Okay. And do they comment on that? Or do they have an ongoing clinical trial?
They don't have an ongoing one that they commented on, but it's like, this is what you should do next. And it's not like we don't have those drugs available. We have anti-PD-1, we have anti-IL that exists in the universe to use.
All right. Okay. Awesome.
It was pretty cool.
We'll see. Let's follow up on that. Okay. So from my side, I also have a pretty interesting. I have two very interesting papers today. I also there are a few others that came up recently, so I will probably present in future roundups. But there was so much to choose; it was insane. So yeah, this paper I thought was super interesting, and it's called "Neo Self Antigens are the primary target for autoreactive T cells in human lupus," and it starts from a phenomenon that I wasn't really aware of. Uh, I was surprised that didn't really have this in my radar, which is that MHC class 2 molecules, so the ones that are presenting antigen to CD4 cells, and of course, are a really important part of the immune response, they have the potential to bind to endogenous proteins, not peptides, actual full proteins. And in certain conditions, they can be kind of taken to the surface and kind of presented. They can present full proteins on the surface of antigen presenting cells. And this has to do with the way in which peptide is loaded on MHC class 2, MHC class 2 molecules, and on the structure of MHC class 2 molecules that is, compared to MHC class 1, is very particular. So MHC class 2 molecules or MHC class 2 receptors are presenting antigen, the presenting peptides that are bigger than MHC class 1. And importantly, for the purpose of this, they are actually composed of two chains, an alpha and a beta, but they're separate chains. And therefore, this kind of results in a groove where the peptide is supposed to bind that is more spacious and more flexible than MHC class 1. And this allows it to present peptides that are usually bigger, like 13 to 19 amino acids. And during the kind of the synthesis of MHC class 2 chains, they, of course, because they're, bound to they're supposed to be presented on the membrane, they start translating, they have a signal peptide, they get transferred to the endoplasmic reticulum, and they continue there and they get, anchored to the membrane. And importantly, during this step, there is a particular protein, a molecule called invariant chain, which is occupies the peptide groove during that step in which the MHC is located inside the ER. And this is important because this kind of is important for the structure, and this also something similar happens with MHC class 1 molecules. They need this something to be occupying the groove in the middle in order to be properly kind of assembled. And importantly, too, and I guess particularly important for MHC class 2 is that the binding of this this chain, this invariant chain, prevents the binding of other proteins that are present in the ER. So this something that was realized many years ago is that if you don't have that invariant chain because you knock it out, then you see all kinds of proteins being binding to MHC and being presented on the surface. Overall, there are less MHC class 2 molecules on the surface because most of them don't get that kind of assembly properly and they get messed up. But of those that do get through the process, they actually can get transferred all the way to the membrane, especially if they end up binding a protein that has some kind of signal peptide or some kind of signal to be transported to the surface without going through an endosome lysosome compartment, which is part of the normal peptide loading in an MHC molecule. So then basically, kind of recap is you have MHC2, they have this invariant chain part during the ER processing. And then once the MHC class 2 goes to the endosomal compartment to get loaded with degraded peptides, as part of the normal processing of antigen presentation, this invariant chain gets replaced by a proper peptide, and that gets sent out to the surface. If you don't have the invariant chain, there are situations in which you can have proteins that have specific, motifs that can bind to these grooves, and this is going to depend on which haplotype of MHC class 2 we're talking about. And they can be presented on the surface. So this is something that actually was not very aware of. So this group, this paper, first authors was published in Cell, 13th of September, came out online. First author is Shunkuse Mori from the lab of Hisashi Arase at Osaka University. And the Arase lab has been kind of exploring when I was doing my research for this paper, I see they have already published many papers in which they show that this aberrant presentation of peptide of proteins has autoimmune consequences on patients on model disease models. So for this paper, they're particularly looking to SLE, into lupus, and kind of long story short, they show that by knocking out, if you don't have this invariant chain, this results in presentation of of proteins from that are eventually get recognized and can initiate a lupus-like disease in mice. So basically, what is very important is they have a mouse model in which they can induce the deletion of this invariant chain in adulthood, and that's very important because you can have a congenital mouse strain that doesn't have it at all. And then these mice have like less CD4 cells, they have less MHC, but they don't develop autoimmunity. But if you, during adulthood, knock out the invariant chain, and all of a sudden you end up having antigen presenting cells that are presenting peptides, internal peptides that are being kind of caught on in the ER. And this initiates a lupus-like disease in these mice that they're studying. So basically, they show that with this tamoxifen inducible mouse model. They show this effect. They show that these mice develop, B cell responses against they develop anti-nuclear antibodies and other self-antigens that are observed in lupus patients. And it's they do a lot of like kind of analyzing the kind of peptides that are presented, and they show that there's a kind of different repertoire of proteins being presented on this MHC class 2 molecules. And so they call these new things being presented, they call it neo self antigens. And this is how kind of they refer to this. So I thought it was very interesting. They show that these mice develop CD4 T cells that are reactive to these neo self antigens in this inducible knockout mice. And they show that if you isolate these CD4 cells from these mice and then you put them in a congenitally knockout mouse, then you can have again is autoimmunity. So these are clones that are recognizing things that are prevented from appearing if you have a proper kind of processing of the antigen. So they see that and so they look into lupus patients, and they also look that they are many of the clonally expanded CD4 cell clones in lupus patients respond to antigens presented by by cells that are lacking this invariant chain, suggesting again that they are recognizing these neo self antigens in lupus patients, and they suggest that this can be up to 10% of the TCR repertoire of clonally expanded CD4 cells in lupus patients, which is a lot. So it seems to be quite, from what they're suggesting, seems to be quite a important factor. And here, I think I thought was also very interesting and very scary is another installment of my ongoing series. Is really EBV is a really crappy virus. They suggest that because there is a kind of epidemiological relationship between reactivation of EBV and this development of lupus, they suggest that there might be a kind of a mechanism by which EBV reactivation can induce these neo self antigen specific cells, because there's certain there's a particular one factor called BZLF1 that is part of the reactivation cycle of EBV that can downregulate the expression of this invariant chain, and therefore kind of mimicking this of what they show in these mouse models. So increasing the presentation of these self antigens, and that in this way you can kind of break this tolerance and generate cells against self antigens observed in lupus. So I thought it was very interesting. I have to say, kind of, this does not seem to. It's hard for me to see how much of a consensus there is around this mechanism, because I don't see a lot of people publishing about it. It's mostly this group, but I think that it's very interesting. The data they present seems very good, and I don't know. It feels kind of makes so much sense. No, from a first read, I really like the story.
So high level, then, just to make sure I follow it right, when an MHC presents a full protein, it doesn't go through the usual ER processing. And is that good? Does that prevent autoimmunity, or does that induce autoimmunity?
So if you have absence of the invariant protein, invariant change from like during thymus development, so like congenital from the start, then you're gonna be presenting those protein antigens from the start. So therefore, in the thymus, you're gonna delete clones that are reactive, right? So then you don't end up having those clones in your repertoire. In principle, that's kind of the idea that they suggest. But if you lose this expression in adulthood and you're presenting all of a sudden the clones, the reactive clones are out there and they get stimulated, then you induce autoimmunity.
Got it. All right. Well, I don't have a segue for but this was a paper that I thought was really cool from the title, and it's really cool to me, but less cool to other people maybe. But it's okay. So I'll tell you why I thought it was going to be extra cool to start with. And so bile acids are really important for immunity and the microbiome and everything else. And so I was there was a really coal new paper that was starting to do some structural and functional analysis of bile salt hydrolase. So the paper is called Structural and Functional Analysis of Bile Salt Hydrolase from the Bison Microbiome. First author, Radwa Assar and Poonam Dewal and Antonio Rizzuri are all first authors, and Antonio Rizzuri is also the last author. It's in JBC, coming out in September. Now, when I think comparative biology, I go back to our shark paper days, and the shark biologist had, I'm like, it's really cool sometimes to get into comparative biology and see what we can learn.
Yes, did you say bison?
Bison. So I'm like, I don't care about shark microbiome as much, but bison could be interesting here. And then secondly, the bile acid milieu that we have in our gut, really, that's actually why we think people just don't get C. diff all the time, is the bile acids inhibit C. diff. And when you take antibiotics, then you get, the bacteria are gone. So now there's a place for the C. diff to grow up and get you sick, or other things to get you sick, because there's no bile acids inhibiting it, because all the bacteria that modify your bile acids and metabolize them went bye-bye. And so there's a really infectious component, and then a component without that then sculpts your immune system, as we hear in various talks and places. And so I'm like, okay, cool. Maybe we're going to get a really cool structure function analysis of a bile salt hydrolase and how that's shaping the microbiome and thus immunity. That's what I going in with. I came out of it with a structural analysis of a bison bile salt hydrolase.
It happens to all of us, Jason. We go for one thing and okay, but what did you learn? Is there anything for us to?
So, different species and different bile salt hydrolases are prone to cleaving off different entities. Bile salts are all conjugated with a glycine or taurine at the end, and so different ones are better at cleaving them, and this is true of this one. It has a very good analysis of the bile salt hydrolase. I have a KD. I have, its properties at different pHs. It has all the things I'd want from the first part of a structural biology paper. What I was hoping for was some comparison. They do some comparison to humans. And I think the conclusion here, and they actually use a pan inhibitor that's kind of come out with this warhead that they were talking about, which I think is hilarious. They talk about it's a suicide inhibitor with an alpha fluoromethylketone warhead. It binds, it kills the enzyme, and doesn't work again, kind of like how aspirin does. They show a couple of cool things from that perspective. That warhead's being looked at as a way to interrogate these systems across species and across systems. So you want to work in mice and people, and it works in mice and people and bison. So that's actually good. It seems to be a good tool drug. And it seems the other point, though, is that it's not great at binding, but it's really good at killing the enzyme. So you have to let it cook for a while. You can't just throw it on as a competitive inhibitor. It's not. It has to bind and do some chemistry, and then that enzyme copy is no longer functional. So when you plan your assays, know that. The last bit, though, is I was hoping we'd get, remember when you were looking at shark immune systems and shark structure, you learned a lot about people from it. I would say that this really just shows us that we're all just vessels for the various microbes in the earth. And that's our job in life, is to eat and poop out them and keep them going. Because fundamentally, they're it has some structural similarities to other things, but it's also its own unique thing, but also has the same enzymatic activity you expect. And they're all pretty dang variable. And there's not a lot to glean about evolution here. It works better for its bugs than the human bugs. But it does all its things you'd expect it to. And essentially, there wasn't some oh man, this explains something about ruminants or herbivores or immunology. It just does its dang job, like all of these do their job in bacteria.
I like that little philosophical tangent you went on. And in the end, what are we if not just containers for millions of bacteria to keep living? off of us.
Yeah, pretty much.
That's pretty nihilistic, but I'll take it. I'll take it.
We all go back to soil one day, and the soil's full of them.
Oh my God. All right. On a, on a a better note, I want to talk about a paper that has to do, you know what? People make choices. People change their circumstances to, you know, bring them closer to their essence. And in this paper, we're going to study a very interesting, I think it's a very interesting study that looks into the kind of the changes that happen in the immune system of people that undergo gender affirming hormonal therapy. So basically, trans men transitioning into from female to male, and they use this kind of this people to have an experiment, basically to see what happens to their immune system. All right. So as I said, the paper is called "Immune System Adaptation During Gender Affirming Testosterone Treatment." The first authors are Tadepalli Lashmikant and Camilla Consiglio. And the corresponding authors are Niels Landegren and Peter Brodin. And actually, I did hear about this story already from Peter Brodin. I think it was at IUIS. I remember this story. I remember thinking, oh, that's pretty cool. So, what is this? What is the setup here? They have 23 trans men that went through hormonal treatment with a testosterone analog, and basically, samples were taking before starting, and then 3 months and a year after the starting the therapy. And I guess one of the main um, motivations behind the study is the fact that we see, of course, that there are differences in the immune responses to infection and to autoimmunity between men and females. And I think one of the most striking examples is the differences in COVID mortality that we observed between men and women. Women did significantly better than men in surviving, severe COVID. And this, in part, this is because they seem to mount a better interferon type 1 response, which protects them from a viral infection. However, the flip side of this is that mostly uh, this often results in more adverse reactions to, for example, vaccination. In the case of COVID, women have um, a higher rate of long COVID, and they are also affected more by autoimmune diseases, particularly lupus that we just spoke about. And so a lot of the question is, where are these differences coming from? Because you have, on the one hand, of course, genetic differences. Women have two X chromosomes, and then you have men with an X and a Y. And there are certain genes in the X chromosome that we suspect have some kind of impact on immune responses. And one of them is TLR7. Basically, it's related to viral recognition of viral infections. And women basically have a double load of TLR7 because it's a gene that it's um, not really silenced in the second X chromosome. So you have basically both chromosomes expressing this gene, and therefore you have more TLR7. And this probably one of the reasons why you have a higher. Now, this is this has been suggested to be the reason why women have higher interferon type 1 responses. Um, but then on the other hand, we also know that estrogens and male hormones, testosterone, progesterone, they also influence the immune system. There are immune cells that have receptors for these hormones. So what is it? So here we have a beautiful natural experiment. People that you can have this take samples through time and see what happens. So I don't want to make it too long, but what in principle, what is that they see? They see that indeed treatment with testosterone, or these people, they start showing no testosterone in their blood. They have a reduction of estrogen and female hormones, so that the transition is effective. And the main differences that they see are, they see clear differences in interferon type 1 responses and also TNF responses. When it comes to interferon responses, they see that trans men that undergo hormonal therapy, they have a reduction in their type 1 interferon response, but nothing related to TLR7. In this case, they show particularly in PDC cells that a downregulation of IRF7, which happens through protein. So they have less IRF7 and more of a protein called SOC3, which suppresses interferon responses. So IRF7 is an inducer of interferon responses, and SOC3 is an inhibitor of interferon responses. And this get affected by the testosterone on the cells. So they have testosterone receptors, and these initiate these changes. And so you basically have a lower interferon type 1 response in these trans men. On the other hand, they see another pathway, which is TNF signaling, and particularly in monocytes. They see that the direct effect of testosterone increases the expression of SLAMF7 receptor that induces a higher TNF response. And this is something that they see particularly in NK cells. So they see an increased NK response, and they have stronger interferon gamma production in NK cells already following 3 months of testosterone treatment, which I think is pretty quick. So I mean, I don't want to go too much in the details, but I think what is very interesting that they show that there are clear differences in the immune responses of trans men. And this is only because of like 3 months or a year of testosterone treatment already kind of rewire the response towards less of an interferon type 1 response, more of a TNF interferon gamma response, which is closer to what you see in cis men, and further kind of really shifting away from what you see in cis women. And I thought it was so very interesting.
All right, that's super cool.
Right?
That's a great way to have that experiment. Oh, that was a good one. I think you got some good papers in this time.
Right? I was super like, oh yes. I was like bookmarking them on Twitter. Like, I'm gonna talk about these papers.
X now, not Twitter.
Oh, you listeners understand what I mean.
All right. Well, we're going to have a great conversation here with Dr. Roslyn Kemp at the University of Otago. But before we get to that, are you looking to improve your T-cell therapy development? With STEMCELL Technologies' new GMP manufactured Immunocolt XF, you can achieve robust T-cell expansion with high cell yield and viability without the use of serum. To learn more, visit www.stemcell.com/immunocolt-XF.
For this episode and the second part of today's episode, we are very happy to be joined by Dr. Roslyn Kemp. She's professor of immunology at the University of Otago in New Zealand. We're calling us from Australia at the moment. We're very excited to have, some down under representatives in the podcast today. And she's going to be talking to us about her research on colorectal cancer, mucosal immunology. So we're very happy to have you on, Dr. Roslyn Kemp. Welcome to the Immunology Podcast.
Thank you for inviting me. I'm looking forward to it.
All right. Well, I guess I'm the mucosal person in the group. So I will start. I first wanted to talk about one of the systems you'd created because I thought this was super cool. And this is from work a couple years ago. You'd done a organoid model in the colon to look at immune bacterial interactions in Crohn's disease. So maybe you could start by talking about the problem in so much as what you were trying to solve by doing this. I think that gets into a good chunk of, hey, what's going on in that system and why would you need to go create something like this to understand anything?
Sure. So inflammatory bowel diseases, obviously Crohn's disease is one of those. And they've been really recognised as really complicated diseases. What people tend to do is try and address each component separately, and that's what we'd been trying to do as well. But actually, what we do know is that there's a host genetic susceptibility, there's involvement of commensal bacteria, there's involvement of immune response, and there's involvement of the epithelium. So we thought if we really want to try and work out what's going on in inflammatory bowel diseases, then we need to try and model the entire system as a whole. And this really reflects sort of the approach that my lab takes, is that if we're going to study complex diseases, we need to embrace the complexity of those diseases and try and look at everything at once. So that was our sort of noble goal. And then we sort of started trying to do this work and really got in the weeds of trying to look at these complicated systems. So this was originally a collaboration with a epithelial physiologist, Grant Butt, and a gastroenterologist, Michael Schultz, and I came in as the immunologist. And between us all, various postdocs and students over several years, we created a model whereby we take crypts from patients, so directly from colonoscopy samples. We can grow those into organoids using protocols established by others. And then we take the blood cells from the same patients, and then we can set up a per patient model where we can look at the epithelium, we can look at the immune cells, and we can add in bacteria as well. And now what we've tried to do is replicate what's going on in the intestinal immune system of people with Crohn's disease. So that's the model, and the goal is then to try and be able to test different therapies on different patients and see if we could try and predict responses to those different therapies.
So in this particular study that you did, you were looking into Crohn's disease and mechanisms of pathogenesis in Crohn's disease. Maybe would you like to share with us a little bit what makes this model a good, so for studying Crohn's disease and particularly epithelial barrier inflammation in the gut? What was the most important connection between Crohn's disease and this model that you saw in this particular study, for example?
So the model is really a two-dimensional monolayer model. So you have effectively a line of epithelial cells, and you can put the immune cells underneath those, and you can put bacteria on top of those. So it looks sort of like how you'd have the immune system working in the gut. What we had tried to do was, first of all, measure how leaky the gut became by measuring the transepithelial electrical resistance across that. But we also started to look at what happened to the immune cells when that epithelial barrier was damaged, and whether it made a difference whether there were commensal bacteria there or not. So the idea was that we would be able to effectively prove a role for an immune cell causing the damage, and that way that would open up a chance for therapies. What we found instead was that every patient is completely different. And so that meant that we didn't get lovely error bars and stars of significance. But what it does mean is that it's an actual real life model of how the disease works. The strength of the model, and what we showed in that first paper, was that it does allow you now to actually look at each patient one at a time. So the weakness of it is that you can't average out a nice result. The strength is that actually you can now go and look at an individual, you can follow them over time, and you can find out what is happening for that person, with the idea being that you can then target different therapies to that person.
So one technical question just for me, because I'm a nerd in this field. Are you using an air-liquid interface system, or are you keeping everything in liquid on both sides the whole time with this model?
At the moment, we're using liquid on both sides, but there have been other studies by other people that have used the air-liquid interface and shown it to be quite effective. So we're starting to try and work out how we can do that as well at the moment.
Yeah, because I'm wondering about anaerobic bacteria and everything in particular in that environment being so critical to the bugs versus just the aerobes that will survive explant.
So what we've had to do is use heat-killed bacteria. Okay, so that's what we've done so far. We've started a new project with a PhD student, Gemma Laws, who's actually moved now to do a postdoc at Oxford, and another student, Millie Roy. And what they're doing is actually trying probiotic strains of bacteria, so commercially available probiotics that we can now test. It would be really nice to look at some more realistic bacteria interactions for each patient. So the long-term goal would be then to take the fecal samples from the patients and isolate those bacteria and do that. So this is all in our to-do bucket. It's quite hard to do because we don't actually have lots and lots of patients in New Zealand, and we also don't have any money. So it does mean that everything has to be scaled down a little bit.
And so also in your lab, this is most thinking about Crohn's disease and the role of the microbiome. But you're also, some of the work in your lab is talking about colorectal cancer and other diseases, that have a very important immunological component. Can you, for example, these models in which you're looking at organoids that have the different layers, are they also useful to studying tumor microenvironments or in the context of colorectal cancer or other cancers of the gut?
I mean, the organoid models are useful in those contexts. We aren't using them for cancer. So it's sort of in the organoid field there. It's more difficult with cancer because they're effectively tumoroids rather than organoids. The advantage of the intestinal organoid model is you've got different types of intestinal cells that are there. It's quite reflective of the whole organ. In terms of our cancer work, we haven't moved into an organoid model simply because we're looking at what's happening in the actual patient tumor samples. So we're not trying to replicate or to manipulate that tumor environment. For colorectal cancer, patients are diagnosed, and then the first thing that happens is that you have surgery and take the tumor out. Okay, so the idea then is to work out for each patient, what is the immune response that's going to work for that patient? Therefore, what is the potential therapy that is likely to work for that patient?
Right, because every patient's going to have a biopsy at diagnosis, and so during the workup, you could theoretically have a companion test to dictate therapy if you can get it to work, right? But you can imagine a 5-day period where you do an organoid experiment and bam, you want this drug, not that drug. Right? Is that basically the goal you're going for?
No, so that's what other people are doing. So it depends very much on where you are and how the health system works, whether you get a biopsy or not. So I think that's a whole other conversation. For us, growing up, cancer organoids isn't really our goal. What we've been able to show is that there are types of T cells that are present in the tumor microenvironment of people with colorectal cancer. We've been doing some sort of high dimensional cytometry to look at the complexity of those immune responses in there. And what you can see in some patients is that they have certain populations of T cells expressing high levels of things like PD-1, CTLA-4, and those populations vary quite a lot from patient to patient. Some of those populations are going to be effector cells that need to be, effectively turned back on, but some of those are also cells that have suppressive function that you definitely don't want to have turned back on. So the goal for our work there is, again, from a individual patient perspective, is to be able to define an immune signature from the tumor of those patients and then do our best to predict what therapy they're likely to respond to. So at the moment, what we have is a lot of immune-based data on this from a whole different set of platforms. And where we're trying to go now is to try and model some of that data so that then we can create a really useful predictive tool. Again, this is what others have been trying to do as well.
So one of the, I think it's not so recent, but you've also been looking into, for example, I don't know if this has any diagnostic, if you're planning on using it as a diagnosis tool, but like high dimensional analysis, like really going, you mentioned you have some in your work, mass cytometry or things like that. Is that, sometimes I think I look at people proposing this, and then I think of the reality of the clinical practice. And then I'm thinking how realistic is to have this very, very detailed or very, work intensive and sometimes very difficult to analyze measurements in order to kind of realize more personalized therapy. What is your opinion on that sense, or your professional view?
So my opinion is that this is feasible in a clinical setting, but it can only be done in a partnership with a skilled research lab. It's not something that's going to be easily done in a pathology lab. You're exactly right. And so what my lab is trying to do now is to set up relationships with our clinical trial organizations and our oncologists to say, include us in the clinical trials. We can do this analysis. We've been trying to develop some simple ways of visualizing this sort of data so that it's interpretable for the doctors who want to know, that they want to know what's going to work for their patient. They don't want to necessarily understand all the ins and outs. And I think people worry often, certainly with clinical trials, that, this is too complicated, it's too complex. But so is the statistics involved in analysing a clinical trial, and nobody worries about that. They just outsource it to someone who knows what they're doing. And I think that's something that we can be involved in as a community, not just obviously just us.
So kind of shifting gears a little, you've also done a lot of work looking at T cells and colorectal tumors, and you talked a little bit about PD-1 and checkpoint inhibitors, and we know that I think the story is well established that microsatellite and stable colorectal cancer, great, it works, it has an indication for it, but everything else has kind of been a struggle. Could you talk about the T cell angle of that? What's going on there that is breaking down, and so that only some very specific types of colon cancer are amiable to immune therapy, and otherwise the T cells are like, peace out, cancer, you can go hang out?
So I think the problem with colorectal cancer is that it's, there's subsets of MSI, stable and unstable cancer as well. It's not quite so simple as one works and one doesn't. I think what we've shown is that the complexity of the T cell response within the tumor is just huge. It also depends where in the tumor you're looking. Okay, so we get a tiny bit of tumor and we mush it all up and we have a look at the cells. We've been doing some whole slide imaging of tumor samples, and the T cells are all in different places, and the phenotype of those cells is different in different places. So it becomes really difficult to do an overall, this is what happens in this sort of tumor versus this is what happens in this sort of tumor. What we did find was that the T cells that are enriched in the tumor compared to the healthy bowel tissue in the same patients, they express markers of activation of ability to kill, but also really strong markers of regulation and suppression. So I had one student who called them activated effector inhibitory receptor regulatory T cells, right, which is everything. So to me, it's going to be, when you combine the complexity of the tumors, individual tumors, and the complexity of individual immune responses, there's never going to be a really simple answer of that you can stratify patients down this road and think this is going to work, and you can put patients down this road and think this is going to work. To me, it is always going to have to come down to an individual patient.
I think using that as a kind of a segue to my next question is, if you're gonna, in the case of New Zealand, if I may have the liberty of saying, it's a small community, I guess, New Zealand, that you're that you are. So when you're thinking about individualized patient care for really small, like, sometimes I feel like always is the economy of scale is always good, because then you can be more kind of effective with your effort and the therapeutic value. But on the other hand, I guess if you have very small communities, you're never going to have this scale. Is this something that you think about when thinking of the reality of, like, treating small communities, or is that something that crosses your mind?
It very much drives a lot of our research. There's a situation in New Zealand and in many other countries is that there's a limited amount of funding for cancer drugs. When the cancer drugs are biologicals, then they're insanely expensive. And so it means that only some of them are going to be funded, only some of them are going to be available to patients. Most people aren't going to have sufficient funds to pay for it themselves. And so in our situation, that targeting of the available drugs becomes really, really important. For example, in inflammatory bowel disease, only a third of patients respond to anti-TNF therapies. These cost in the tens of thousands of dollars per year. If the government is funding that, we need to make sure it goes to that third of patients that are going to respond. And I think this is a reality for many countries, particularly those that are resource limited, it really is about making sure the right people get the right drugs that they need that's going to have the best effect.
Just want to say that sometimes when you think about certain biologics, especially antibodies, they're actually so cheap to make. It's no licensing cost that you're paying. But well, never mind.
Well, yes and no. The sterile manufacturing is hideously expensive. That being said, having been on Humira and having failed Humira, and having been involved in research trying to figure out what would to discern responders from non-responders for anti-TNFs, it still seems like we're not there. So what do you think? Like this has been a goal, not just of, a place like New Zealand where they don't have, where there's that cost issue, but even in the US, insurers are like, why am I spending this money on someone where it's not going to work? And we've not gotten there despite everyone trying. So what do you think the barriers are for that? Where do you think there's something fundamentally wrong in our approach? Do you think it's just a really hard problem?
I think it's these sorts of drugs treat complicated diseases. So that's the first thing, is it's not simply, it's not simple. Chemotherapy is never simple, but the point is you're not trying to go in and just kill something. You're trying to go in and modulate the function of multiple types of cells in different people at different time points, depending what the disease is and how it's going to work. So there's always going to be a bit of a hit and miss. What we have been trying to do with the inflammatory bowel disease organoid model is to try and predict responses, and we can, in a very small study, we can show that we get some repair of that epithelial barrier with anti-TNF in some of the patients that are responding. Where we're going now is to try and work out whether the immune response in those patients is also predictive of a clinical response. And then the idea would be that you sort of try and combine some of these parameters together to see if you could make the response. I think it's also we've sort of spent a bit of time talking about cost, but I think actually it's worth thinking about the sort of patient life cycle of these drugs too, is that generally a patient, certainly with inflammatory bowel disease, is going to go through a whole lot of trial and error to see what's going to work. And that can take months and years of pain and discomfort and disruption to quality of life. So even aside from the cost, this is something that we need to be thinking about, is that we need to better serve the patients as well.
So I think shifting gears a little bit, I wanted to ask you a little something about your side, or maybe it will be a side, but another endeavor that you have recently embarked on, which is writing books about scientific education and what it takes to be a good scientist. And I thought that was very interesting. We have some other guests that also have kind of spoke about, inclusivity or helping, new generations find their footing in science. And I think it would be very nice to hear from you. How was your experience? What motivated you to write this book? Maybe you can briefly comment on the content, and what would be kind of a few highlights that you think our listeners would like to hear.
Okay, so first of all, the book was written with a co-author, Deborah Brown at the Trudeau Institute. So Deb and I actually postdoc together 20 years ago now and maintained a friendship, and both of us maintained an interest in education. So my first point is that, the people you meet early in your career end up being lifelong collaborators as well as friends. So what Deb and I wanted to do was really, it started a few years before COVID, which was really thinking that the general public don't really understand how science works. During COVID, there was this much more obvious mistrust in science and misunderstandings and how to look at data and how to look at evidence. And so really what the book is it's a textbook designed for undergraduate students studying science. And it goes through how science works, right? It goes through experimental method, hypothesis design, hypothesis generating research. It goes through how publication works. It goes through how peer review works, how people get funding, how careers can work. There's a chapter about the importance of statistics in science. And so it's really designed to get people to understand how science works and to improve sort of overall scientific literacy, not necessarily about scientific facts, but actually how to interpret data, how to think about it. And so that was the goal, was that, in theory, my sister or my dad could read it, who aren't scientists, and think, oh, this is what she does. But also that, the media can read it and be like, oh, okay, so this has actually been through these rounds of peer review, which people do for free. They're not, being paid for this. And it sort of shows that the quality of data, of scientific data, is important. In terms of one of the chapters I think we're most proud of is the last one, which is really about the additional skills that you get when you train to be a scientist. So things like creativity and time management and budgeting, and how many careers there are with a science degree and how skilled you can be, and that the job of a science graduate is often not science, but it's many other things. And I think I'd like people to see that it's a really highly skilled profession. It doesn't mean you need to be in a lab.
Excellent, excellent. And well, and to wrap this up, we typically end with a science adjacent question, but I think it's going to fit right into this really well, which is, what is the best piece of advice you've ever gotten, either professional or personal?
So certainly professionally, the best piece of advice I got was to ask lots of questions. So you start a new job, you ask everybody, you go to a talk, you ask the questions. This is something I've tried to get my students to do, is it's like, you are not the only person who doesn't understand. It's completely fine to be the brave one that asks the question. And it can only, people aren't mean. Even in a big scary conference, you can ask a quite simple question and most of the audience will be pleased you asked it, and you can always get something from it. And if you don't know, then you need to ask and people will tell you. That would be my main thing. I don't know if that works in a personal thing. Probably. I mean, yeah, you need to know if you're sending your kid to school that's going to work, I suppose.
Yeah. Or people are shy to ask questions, and sometimes they do. Absolutely.
I think that's good advice. But I mean, but it's really hard, right? You go into a conference and then you see everybody's, nodding their heads and you're like, oh, well, I guess this is very obvious to people on my left, to people on my right. Should I make a fool of myself making this question? But I guess in the end, it's true that it has happened to me that people then came up to me and said, oh, thank you. I was trying to make that, find the courage to speak up, and you kind of took that out of my shoulders. So I guess it's always good to be the one looking for more answers. All right. So thank you so much, Dr. Roslyn Kemp from the University of Otago Otago in New Zealand. So now you said you are you're having what's the word? A sabbatical. Sorry, sometimes my English doesn't English. In Australia. So how is that common for academics in New Zealand to? I guess you have very close ties with Australia in general.
Yeah, so I work with a lot of people in Australia. We meet up at conferences all the time. A huge amount of my work is done using mass cytometry, and we don't have one in New Zealand, so we have to come to Australia to do our research. And so I'm in Perth working with some really cool people doing some really good cytometry-based research. And so, yeah, for New Zealanders, it's important to get out into the world a bit. We've had a break with COVID, so it's been really nice to be exposed to some new ideas and some new people, and some sunshine, which is really good. Yeah.
You don't get much of that in New Zealand.
No, well, no, not in the bit where I live. Yeah. Lots of seals, not a lot of sun. Yeah.
I see. Okay, but in Perth for sure.
Yes.
Well, thank you again for coming on.
Thank you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.